Hello students, today I am going to teach about the structure of nephron. Okay, structure of nephron. In each kidney has 1 millions of nephron. Okay, so we have the one pair of kidneys. Already we have learned in previous video about excretory system. So kidney structure is also we have learned. Okay, so each kidney has 1 millions of nephrons. So nephron is, it's a structural and functional unit of kidney. So total kidney is made up of nephron. So each nephron is a structural and functional unit, means actual blood filtration processes takes place in the nephron. This is the structure of one nephron. So actual blood filtration processes takes place in the nephron. So that's why these are structural and functional unit of the nephron, structural and functional unit of the kidney. So we will discuss about the structure of nephron. So nephron has two parts. So basically the nephron can be divided into two parts. They are Malpighian body and renal tubule. Okay, Malpighian body, renal tubule. Next, Malpighian body again it divides into two parts. One, Bowmans capsule and glomerulus. So here this structure, so about this structure is called Malpighian body. Here this structure is called Malpighian body. Next, this tube-like structure is called renal tubule. Okay, so we will discuss about Malpighian capsule. First of all, Bowmans capsule. So here Bowmans capsule. Okay, so this cup-like structure is called Bowmans capsule. So here I am drawing here again. So this cup-like structure is called Bowmans capsule. Okay, here it made up of two layers. Okay, it's made up of two layers. The inner layer is called, here inner layer is visceral layer. This one outer layer is parietal layer. So here this inner one is visceral layer, outer one is parietal layer. So in between these two, cavity is present. Okay, so here the visceral layer made up of a special type of cells. They are called podocytes. So here the visceral layer made up of a special type of cells. They are called podocytes. Here this is the inner layer. Here this inner layer made up of a special type of cells like this. Okay, so here the visceral layer has like this. They have some pseudopodia like structures are present like this. These cells are present are like this is one cell, this is one cell. So these cells are called podocytes. So in between the podocytes, here some gaps are there. Some gaps are present. Here the cells, each cell cannot attach to each other. They leave some gaps. These gaps are called filtration slits. The gaps are called filtration slits. Okay, these gaps. So in between the two cells, here some gaps are present. These gaps are called filtration slits. Okay, so this is about visceral layer. Next, parietal layer. Parietal layer has simple squamosal cells. Here onwards it has simple squamous epithelial tissue. Okay, so it's made up of simple squamous epithelial tissue. Which one? The parietal layer. But visceral layer has a special type of cells. They are called podocytes. The podocytes have some slits, some gaps are present. These gaps are called filtration slits. Okay, so this is about Bowman's capsule. Next, glomerulus. Next about glomerulus. So here, in this cup-like structure, the efferent arteriole, this is one efferent arteriole. So in each kidney, so whenever, for example, here this is the kidney. So here this concave surface is called hilum. So through the hilum, the renal arteries enters into the inside. Next, renal vein comes out. So renal artery, when renal artery enters into the inside, divides into many branches. Again these branches divides to form finally a small minute capillaries. These are called efferent arteriole. So each efferent arteriole enters into in each nephron. Okay, so this is the efferent arteriole. Whenever it enters into the Bowman's capsule, here this is the Bowman's capsule. So this cup-like structure, whenever it enters into the inside, it divides into many branches again. So these branches, they rejoin to form efferent arteriole. So these branches, it divides into many branches, many small branches called capillaries. So this capillaries are called glomerulus. Next, glomerulus comes out, or glomerulus comes out through this efferent arteriole. Okay, efferent arteriole forms the glomerulus. Next, from the glomerulus, the blood passes away through the efferent arteriole. Okay, efferent arteriole and efferent arteriole. Okay, so here this glomerulus, here this is capillaries. Here the glomerulus, the wall of glomerulus made up of simple squamosal epithelial tissue. Squamous epithelial tissue. So for example, here I am drawing the single layer. For example, these are the simple squamosal epithelial layer, the cells of glomerulus. Here these are cells of glomerulus. Next, these podocytes are present. Podocytes, the cells of Bowman's capsule. So like this. Next, in between these two layers, here some gap is present. So in this gap, the basement membrane is present. Here in between, basement membrane is present. So here this layer is called filtration membrane. So this one is called filtration membrane. Okay, so filtration membrane is made up of 3 layers. One is glomerulus, the cells of glomerulus. Next, basement membrane, the cells of podocytes or Bowman's capsule. Here the glomerulus cells, the squamosal epithelial cells, endothelial cells also has small gaps. These gaps are called fenestrations. Okay, so here the filtration processes takes place through the fenestrations and also this filtration slits. Okay, so this is about structure of Malpighian body. Okay, so Malpighian body has 2 parts, one Bowman's capsule, second one is glomerulus. So Bowman's capsule is a cup-like structure like this. This one is a cup-like structure is called Bowman's capsule. It made up of 2 layers, double layer. The inner layer is called visceral layer. This inner layer is called visceral layer. Outer layer is called parietal layer. This outer layer is called parietal layer. Visceral layer made up of podocyte. A special type of cells are called podocytes. It has some slits. They are called filtration slits. Filtration slits are present. Next, the parietal cells has simple squamosal epithelial tissue. Next, glomerulus. Glomerulus, it is formed by the efferent arteriole. Efferent arteriole enters into the Bowman's capsule. It divides into many branches to form many capillaries. Then these capillaries again rejoin and comes out through the efferent arteriole. The glomerulus made up of simple squamosal epithelial tissue. So this simple squamosal epithelial tissue is also has some gaps. These gaps are called fenestrations. Okay. So here the simple squamosal epithelial tissue and podocytes, in between these two a basement membrane is present. These three membranes are called filtration membrane. Okay. So this is about Malpighian tubule, Malpighian body. Next, renal tubule. Next, renal tubule. So the Bowmans capsule next opens into a tube-like structure is called renal tubule. Okay. So renal tubule again has different parts of the Renal tubule has different parts. First one, PCT, proximal convoluted tubule. Proximal convoluted tubule. So this one. So here this one is called PCT, proximal convoluted tubule. Proximal means it present near to Bowman's capsule. C, convoluted means here it is highly folded. T means tubule. So tube-like structure, that's why this is tubule. So proximal convoluted tubule. Next, it's made up of simple cuboidal epithelium. It's made up of cuboidal epithelium with brush border. Okay, and also it has plenty of mitochondria. Okay, so it's made up of cuboidal. Here this one is, it's made up of cuboidal cells, cube shaped cells, cuboidal epithelium with brush border, which means it has many microvilli. For example, like this. This is the one cell. Next, like this. Okay, so each cell has many microvilli. So that's why it has brush. It looks like brush. So that's why this is called brush border. Okay. Next, it has plenty of mitochondria. So why it has plenty of mitochondria? Here mainly active reabsorption processes takes place. Active reabsorption. It for active reabsorption. So normally in PCT the reabsorption is highly major reabsorption processes takes place in the PCT, proximal convoluted tubule. So here mainly the components glucose, amino acids, they are absorbed by the active transportation. So active means which needs the energy. Energy is essential for active transportation. So that's why the mitochondria provides a lot of energy, and also they have brush border. So brush border, they increase the surface area, absorption surface area. That's why this is brush border and active and plenty of mitochondria are present. So this is about proximal convoluted tubule. Loop of Henle. Next, proximal convoluted tubule. The PCT opens into the loop of Henle. So this is a hairpin structure, hairpin shaped structure. Okay, so it's long tube-like structure. It is just like hairpin structure. Next, it has two tubes. The descending limb, which one is going to down, that is called descending limb. Which one is going to up, that one is called ascending limb. Okay, so it has two tubules. First one, descending limb. Okay, it's going to down, so it moves to down. So that's why this is called descending limb. So this descending limb is also made up of cuboidal epithelial tissue. Next, it is permeable to water. It is permeable to water, impermeable to salts or sodium chloride. Okay, it is permeable to water, which means it absorbs the water but does not absorb the sodium chloride. Next, ascending limb. Next, ascending limb. So this one is ascending limb. So this descending limb is slightly thinner the wall of, okay. Ascending limb, it is also made up of cuboidal epithelial tissue. Next, it is permeable to salts, sodium and chloride, and impermeable to water. Okay, so opposite, both are opposite in nature. Okay, it is thicker. The wall is slightly thicker. Okay, so this is about loop of Henle. So loop of Henle is its hairpin structure. It has two limbs, descending limb and ascending limb. So descending limb, both are made up of cuboidal epithelial tissue. Descending limb permeable to water, ascending limb permeable to salts. Okay, so this is about loop of Henle. Next, distal convoluted tubule. Distal convoluted tubule. Next, so distal convoluted tubule, which means, so the distal means it away from Bowmans capsule, that's why this is called distal. Again, convoluted means it is highly folded structure, that's why this is called convoluted. So tube like structure, so tubule. Distal convoluted tubule. This is also made up of cuboidal epithelial tissue, but without brush border. So without brush border. So which means here the microvilli are absent. Okay, here only less reabsorption processes takes place. So that's why here mitochondria are also less mitochondria are present. Okay, next, collecting duct. So finally the distal convoluted tubule. So this distal convoluted tubule opens into the collecting duct. So collecting duct opens made up of columnar epithelial tissue. It's lined by columnar epithelial tissue. So next, so all collecting ducts. So here, so for example, here this is the one nephron, one more nephron. Distal convoluted tubule. So all distal convoluted tubule opens into the collecting duct. So all collecting ducts remaining in, for example, in pyramid. So in the kidney, the medulla shape of structure is called renal pyramids. So in the renal pyramids, all collecting ducts are joined. All collecting ducts joined to form a long duct, that is called duct of Bellini. Okay, all collecting ducts join to finally to form a long duct. This one is opens into the in papilla region. So papilla region, they are opens into the, in papilla region opens into the pelvis. So this large duct is called duct of Bellini. So all distal convoluted tubule opens into collecting ducts. All collecting ducts in the pyramid of one renal pyramid, all collecting ducts opens into the a duct of Bellini. So duct of Bellini opens into the pelvis near papillary region. Okay, so this is about structure of nephron. Okay, next, peritubular network and vasa recta. Peritubular network and vasa recta, which means here in the glomerulus, the efferent arteriole forms the glomerulus. Then after that, the efferent arteriole is it forms, the efferent arteriole is comes out through the glomerulus. Next, this efferent arteriole, it divides into many branches. These branches, they spread they covers the PCT and DCT. Okay, it forms the some branches. So these branches that a network manner. Okay, so the different arteriole divides into many branches. That branches covers the PCT and DCT. So they are called peritubular network. Okay, so from this network, one tubule, it's run along through the, it's present along through the this loop of henle, and also it has again junctions also like this. Okay, so this some tubes arranged along through the loop of henle. So these tubes are called vasa recta. So here these are called vasa recta. So in the vasa recta, the blood flows through the vasa recta. So first of all, through the efferent arteriole, blood enters into the inside. Efferent arteriole has larger diameter. Okay, efferent is smaller in diameter. Means efferent arteriole larger in size, efferent smaller in size. As a result, some pressure is developed in the glomerulus. Okay, so that pressure is very helpful for the filtration process. Next, after that efferent arteriole it divides into many branches, forms a network manner. So that network manner here, this network manner is called, that network is called peritubular network. From this network, some tubes are arranged along the henle loop. So they are called vasa recta. Okay, so this is about peritubular network and vasa recta. Next, types of nephrons. Next, types of nephrons. Types of nephrons. So nephrons generally they are divided into two types. Okay, they are cortical nephrons, medullary nephrons. Okay, nephrons can be divided into two types. Okay, so for example here, for example, just this is the cortex region, this is the medulla region. So kidney can be divided into two regions. Outer region is called cortex, inner region is called medulla region. So first of all, cortical nephrons are not medullary nephrons, juxtaglomerular nephrons, juxtamedullary nephrons, cortical nephrons and juxtamedullary nephrons. So the kidney can be divided into two regions. Cortex, outer region is called cortex, medulla region, inner region is called medulla. So here in this juxtamedullary nephrons, so for example, this is the nephron structure, Bowmans capsule, next PCT, proximal convoluted tubule, next henle loop of henle, next distal convoluted tubule, next collecting duct like this. This is the juxtamedullary nephron, which means here the henle loop is very long. Loop of henle is long and it is extend in the medulla region. So the Bowmans capsule, PCT, DCT are present in the cortex region. PCT and DCT and Bowmans capsule are present in the cortex region. So this type of nephrons are called juxtamedullary nephrons. Normally in human being, these juxtamedullary nephrons are present. Cortical nephrons means here the Bowmans capsule, next proximal convoluted tubule. Here henle loop is very short. So like this. So here these are cortical nephrons. nephrons, which means here henle loop of henle is absent. Loop of henle is very short. It's very short. Okay. So the loop of henle is not extended into the medullary region. So it's very too short. So Bowmans capsule, PCT, DCT are present in the cortex region. So here in the medullary region, this henle loop is absent, not extend up to medullary region. But in juxtamedullary nephrons, the henle loop is very large, okay, very long. It extends into the medulla. So that's why in human this juxtamedullary nephrons are present in the human. So these are help for the production of high concentrated urine. Okay. So these are the two types, cortical nephrons and juxtamedullary nephrons.